research examines how colitis, an inflammatory condition of the colon, impacts liver health in individuals suffering from primary sclerosing cholangitis (PSC), a chronic cholestatic liver disease. We discovered that inflammation in the colon surprisingly helped to protect the liver by reducing the damage caused by bile acids, which are normally produced by the liver to aid lipid digestion. This could change how we approach the treatment of liver disease, advocating for strategies that consider the health of multiple organs together. Our research, as you may know, that the strong association of inflammatory bowel disease, IBD, and PSC has been known for decades. However, the molecular mechanism of colitis may affect cholestatic liver disease remain completely understood. Here we used an IBD PSC mouse model to study the impact of colitis on cholestatic liver disease, and very surprisingly we found that intestinal inflammation and the gut barrier impairment could improve acute cholestatic liver injury and result in reduced fibrosis in a chronic colitis model. This phenotype is mediated via the hepatocellular NF-kappa B activation by gut bacterial translocation, and our findings challenge the previous assumption about how intestinal disease like colitis may affect liver health. Traditionally, leaky gut and inflammation are seen as harmful, but in our studies, we show that under certain circumstances, it can actually be beneficial. I think there are several very important implications in the field of medicine, particularly in the understanding and treatment of PSC. Firstly, given its autoimmune inflammatory nature of PSC, one could consume that anti-inflammatory or immunosuppressive agents could be beneficial. However, our findings, alongside with the fact that anti-inflammatory treatment has been generally failed for PSC patients, suggest that only suppressing inflammation without considering its potential protective role may be detrimental. Therefore, future treatment should consider combining the anti-inflammatory agents together with the drugs which suppress bile acid synthesis, such as FXR agonists. Based on our current findings, yes, we plan to conduct further research to better understand the mechanisms behind the protective effect of colitis on liver disease. We are currently exploring the spatial inflammatory patterns within the liver. We are seeking to unravel the paradox of liver inflammation, understanding why and how it can be beneficial in certain scenarios, such as in the context of PSC. And I think this can significantly advance our understanding of inflammatory disease and their management. I would advise myself to be more open to unexpected results and embrace the complexity of biological systems. The most unexpected results often lead to the significant breakthroughs, so it's very important to maintain an investigative and flexible approach across the whole research process. And there are indeed several aspects which left room for further investigation. One technical limitation is that the IBD PSC mouse model we used in this study doesn't perfectly mimic the intestinal phenotype observed in the human PSC IBD comorbidities. Additionally, our study didn't dissect the effect of gut-derived bacteria on hepatocytes, were direct or mediated indirectly through the immune cells in the hepatic microenvironment. And lastly, most of our conclusions are based on the murine data, which while promising must be considered or approached very cautiously when extrapolating to human physiology. However, in general, these limitations are stepping stones guiding our next steps. I was supported by a scholarship, and this specific research was funded by the Junior Principal Investigator Program of RWTH Aachen.